My talk is about the soft and flexible bioelectronics for the stem cell engineering and the brain-machine interface. My lab at Harvard are very interested in understanding how those neurons in the brain the function of the neurons in the brain give rise to the behavior, how the malfunction of those cells give rise to the disease, or how the different type of cell self-assemble into a piece of a functional tissue. Be more specific, we are interested in converting those high-performance nanoelectronics into the flexible and soft bioelectronics for the tissue interface, and then further integrate those technology with the spatial omics technology to look at the tissue function and properties from the different angle. So before, like I really introduce the technology platform that we developed to address those questions. The first, I want to discuss what are the challenges that we try to address by developing those technologies. The first one is that we feel that to really build like an effective like a tissue electronics interface, especially for example for the brain machine interface, the single cell activity across the entire 3D brain really matters. So this on the left, that is a functional MRI imaging of a human patients, and those red color basically label where the neuron that is firing that define this patient's brain internal state. You can see that the brain really need to recruit the cell activity that is distributed across a 3D volume of the tissue to define those patients' brain internal state. And on the right, there will be more zoom-in imaging, that is a calcium imaging of a zebrafish, a species with a transparent brain at its larva stage. And each dot basically label one neuron that is under firing. You can see that this fish really recruit the single cell level activity across the entire 3D brain to define its behavior state. But if we really want to build the technology to capture the single cell level activity across the entire 3D volume of the brain over time, then we'll face a second challenge in neural engineering that we call it a spatial temporal dilemma. So the individual neural activity, the action potential or a spike that is only lasts for 2 to 3 milliseconds, confined into individual cells at the spatial scale from 10 to like 100 micrometer. But we all know that our brain does not operate at a millisecond time scale, right? It takes days, months, or years to build up cognition, memory, emotions, and recruit a large number of the neurons. Like for the frog, have millions of them, and for the human, there will be 86 billion of the neurons distributed at a spatial scale from centimeter to even meters. And if we want to capture those cellular activity at a large scale over a long time, the ultimately like the bandwidth that we need to build, the bandwidth of technology that we need to build is around like hundreds gigabytes per second to even thousands terabytes per second and need to last for a long time. And give just a direct comparison that the 720p movie that we watched on the YouTube, that is only like a 2.5 megabytes per second. So there's still a huge gap between where we can do now to actually what we can do in activity or tissue activity decoding. That within the tissue, for example, the brain, there are so many different type of the cells. The different type of cells will have a different gene expression, different morphology and connectivity, and collectively they give rise to the different electrical behavior. So ultimately, when we capture the cellular activity and want to decode them, we also need their like a cell type, like molecular information to decode the cell type specificity. So why we think right now is really the golden era for us to address those questions. Because on one hand, driven by the Moore's law, the number of the transistor on one integrated chips that is already goes to the number that is equally to or even higher than the number of neurons in the human brain. And on the other hand, those nanotechnology already miniaturize the electrical detectors and the stimulators down to subcellular size. That in the in vitro setting, we already can provide this stable interface with individual neurons. So on one hand, that is our like a human brain with like billions of the neuron. And on the other hand, that will be our like a powerful computer chip with billions of the transistor, and each transistor can operate at a speed that way beyond the speed of the neural computing. What we are missing right now is really a cable, a scalable interfacing technology can bridge the electrochemical impulses in our like a human brain to our powerful hardware to decode them. So then what is the requirement for this scalable interfacing technology? So I summarize it here. So X and Y, that is the time and the spatial scale. With this two dashed line, basically label the resolution that the technology need to achieve in the single cell, single spike spatial temporal resolution. And we also have this kind of a third dimension that is the cell type specificity that need to be incorporated. And for the traditional bioelectronics, it's very easy for them to achieve the single cell, single spike spatial temporal resolution, but their performance cannot last for a long time. For example, you think that and they cannot very easily to distribute across the 3D tissue. And also it's very hard to incorporate the cell type specificity into this platform. But then you may have the question that since we already have such powerful chip manufacturing technology, why not we just use those technology to build those kind of high throughput, high density electronics to look at the brain activity? So people have already done so. So this is the technology named the NeuroPixel. That is really this using the nanofabrication, this to fabricate these large-scale sensors onto each individual brain probe using the CMOS-driven multiplexing circuits that you can look at thousands to 10,000s of the electrode at the same time. Put those neural pixel probe into the brain, immediately you can capture the cellular activity at 100 to even thousands of the neurons. But the problem is that their performance cannot last for a long time. Like typically, like over one night after the animal have a sleep, you will lose the signal that you can record in the very beginning. They will quickly decay that over the time you won't be able to capture the number of the signal that you can record in the very beginning. So in the first part of my talk that I really want to introduce you how our like efforts to build this tissue-like nanoelectronics that conquer the issue to provide the long-term stable electrophysiology at a single cell resolution. So before I introduce that, we first need to understand what is the challenge that really prevent us from building this long-term stable neural interface in our biological brain. Turns out it's really coming from the size and the mechanical mismatch between the brain and the electronics. Uh, like a biological brain, a living brain is very soft, like a tofu, right? But electronics, on the other hand, is very rigid, like a blade, silicon blade. If you put the electronics into the brain and the human brain or animal brain is constantly moving, it will cut the brain at the micrometer scale and cause this significant drift of the sensors or probe inside the brain. And even you can record the activity from those brains stably, you won't be able to record activity from the same group of neuron over time. And it will also trigger this significant immune response from the brain tissue to the implantable probe so that over the time, you will have a degradation of the neurons and a strong proliferation of the immune cells at the implantation site. So you will lose the single unit action potential that you can record in the very beginning over the long-term implantation. For the patients with deep brain stimulator implanted every couple of months of a year, you need to relocate the stimulation location to find the web neuron to do the stimulation. So when we have this, when we look at this question in the very beginning, it's almost like 10 years ago, we asked ourselves, like if we look back the history of the mankind, like for the material science development, is there any material system that people have developed have already bypassed this size and mechanical mismatch with the biological system? Turns out that there's already have this kind of like a man-made material that can do that. That is this so-called synthetic tissue scaffolds for the regenerative medicine. It's like a 3D microporous soft material that can support the cell grow into this matrix. And when they grow into a piece of functional tissue, we can transplant them back to the biological system. It will not trigger this severe immune response or chronic mechanical damage. So that in the very beginning, we are thinking that why not we build the electronics by mimicking those kind of synthetic tissue scaffold. Also have a 3D microporous structure, subcellular feature size, and the tissue level mechanical property. But most important, to enable the technology to be scalable, that means like a couple or hundreds of the sensors that you build in the lab can directly translate into the industry to build like thousands to millions of them, that this electronics still need to be compatible with the state-of-the-art photolithographic fabrication, a scalable fabrication technology. But turns out that in the very beginning, we cannot even make the tissue level flexible, tissue level soft electronics at all. Because for all the material that can be used for the nanofabrication, such as SU8 or polyimide, that is a dielectric material, or gold, silicon, and platinum, that will be the like metals, the conductors, they have the Young's modulus at least 6 orders magnitude higher than that of the biological tissue. And there's some those kind of elastomers like PDMS or SEBS. They have this tissue level soft mechanical property, but they are not compatible with high resolution nanofabrication at all. So in the very beginning, we can only make a tissue level flexible electronics because the flexibility is not only determined by the Young's modulus, the mechanical property of the material, but is also determined by the geometry of the device, that we can using those rigid material but make them into a very thin structure to enable tissue level flexibility. So this is what we have done in the very beginning. That is, we using those kind of dielectric material, 400 nanometer rigid dielectric material, sandwich the metal to 20 nanometer metal in the middle, made them into this kind of mesh-like structure. You can see how flexible they are, that they can easily be injected into the biological system even through this syringe injection. Like over the time course of development, we have further improved the technology by using the polymer microneedle to deliver those kind of unfolded mesh electronics across the 3D brain. And you can see that by precisely control the implantation, we can distribute this red color labeled mesh-like electronics across multiple brain regions. And those green colors and the purple colors label the neurons and the astrocytes respectively. You can see that over this long-term implantation, this is 6 weeks of implantation, there's minimal degradation of the neurons and the proliferation of the immune cells at the interface with those flexible electronics. By using this like a stable interface, we can capture those neural activity from the freely behaving animals. So this spike, this continuous measured from a freely behaving animal over 14 months, like from its like implantation after it has been naturally died. And you can see that how stable that we can capture those signal. Like for the each channel, we can stably capture those single unit action potential. And after doing the spec sorting, it's not just the amplitude of the signal that seems to be stable, but the waveform of the signal also shows stable, which suggests that we are not only stably capture the cell activity from the neurons over a long time, but maybe we capture the cell activity from the same neurons over this like almost a year-long implantation. To further prove our hypothesis, we further design this high-density flexible electronics array that is for the each like neurons, they will have a high-density electro array to capture their cell activity, allowing one neural activities can be simultaneously captured by multiple electrode in this high-density electro array. And due to the distance between the soma, the cell body of the neurons, to the different electrode, the signal from each electrode will show a distribution of the amplitude. So that over this long time recording, this is a 7 months long continuous recording, that is not just a waveform is stable, but the waveform distribution across a different electrode for the different neurons are also stable. To further prove that we can capture this activity from the same neuron, we also test this kind of neuron representation stability from the orientation-specific neurons by using this kind of dynamic grating with the different angles projecting into the animal's eye and looking at the cellular activity from the visual cortex. We can find that those representation of those orientation-specific neuron to different like a dynamic grating are also stable over the long term, like a 7 months long recording. So together we show that by designing the electronics like the tissue with a tissue-like property, we can enable this long-term stable electrophysiology from the same neurons in the behaving animals. Then the second question is that how we further distribute those sensors array across a 3D brain tissue. The one direction that is currently that is pursued by, for example, the Neuralink, that using the same technology, using the rigid material, but making them into a very thin structure to enable this kind of stable recording. So, but because they still have to use a rigid material, so that what is the limitation? That is you lose the scalability. For each brain probe, there's only limited number of the sensors that you can incorporate. For example, 8 to 16 sensors. So that if you want to, for example, implant thousands of the channels into the brain to look at the neural activity, you need to implant over hundreds of the brain probe. So that they need to have this like automatic implantation machine. And the real solution is that we need to build the flexible electronics like the neural pixel, high density electro array distributed across one brain probe, so that you just need to implant a few electrode, high density electrode across a 3D brain tissue, you can capture large number of the neurons. But because we are limited by the materials, while the interconnects, those electrode can use nanometer thick gold or platinum, it will not contribute too much to the mechanical rigidity. But the dielectric material, that is the material used for encapsulate the channels among from the crosstalk or prevent the leakage current built from those each channel to the solution, cannot be further reduced. As a result, if you scale up the number of the sensors that you can incorporate into those brain probe, essentially you need to incorporate a large number of dielectric material that make the entire probe thicker again and become rigid. And all those issues, the immune response, foreign body effect, the mechanical drift will comes back. The only solution is that we need to bring this tissue level soft material for the nanofabrication, reduce the Young's modulus of the material so that we can enable high density electroarray design with brain level mechanical property. But why the soft material cannot really be used for the nanofabrication or for the brain machine interface? There are two major limitations. The first one is that when a material is soft, what enables the softness, that is the glass transition temperature is lower than the room temperature. You can consider them as cross-linked liquid at room temperature. Or they actually have this micron porous structure so that they can enable the stretchability. But this makes them very easy to be swelled by the water and organic solvent, and which has been widely used in this multi-layer nanofabrication. So that is also patterns defined by the lithographic fabrication will easily be destroyed by the different solvent used for the different layer of the nanofabrication. To solve that, we really need to have the soft material that is a solvent resistant, chemically orthogonal, and photopatternable. The second challenge in that is when the material is soft, because they are really a cross-linked liquid at room temperature, the ions in the biofluidics can easily penetrate them. So that over the time, you will see this significant ion penetration and cause a reduction of the dielectric property, so that it will become the ion conductors over the time, that they cannot further function as a dielectric material for the device encapsulation. So that we also need to have a long-lived elastomer in the physiological condition. So if you imagine a material that is a mechanical soft, strongly repel both aqueous, oil, organic solvent, and ions. From the chemistry perspective, there's only one type of material that can fulfill all this requirement, that is a perfluorinated elastomer, replace all those hydrogen atoms with the fluorine atoms. So my previous advisors at Stanford, Zhenan Bao, introduced me this material that is a perfluorinated polyether, and then I have my student test the material in the biofluidics over 2 years and compare to the standard dielectric material that is widely used for the flexible brain-computer interface, the SU8, it shows the same level of the stability. And this material can be made into the brain-level mechanical property, extremely soft like a structure. And we can further engineering it as a photoresist. We can use a photolithography to pattern their structure at high resolution. So you can see that we can do this multi-layer high-density electrode implantation with brain level, still maintain the brain level mechanical property. We can implant the probe into the brain, and it will, over the long-term implantation, it will have the minimal immune response. We can capture the activity from the same neurons over the long time. But you say this technology is really a scalable technology that we cannot just like using the technology build like a couple of the sensors of tens or hundreds of the sensors. We really need to like further incorporate this technology as a scalable technology to build like thousands to millions of the sensors to interface with the brain. But this is really challenging to be made in the academia lab. That if I ask any of my students, they fabricate 1 million sensors for me in the Harvard CNS, I'm sure that no one really want to do the PhD like that, right? So that we have to translate the technology into the semiconductor foundry using the automatic fabrication pipeline to integrate the large number of the electrode to the brain probe. And we also need to design, because we have this high density, long-term stable interface, we also need to design the new ASIC chips to capture and amplify the signals, as well as a new algorithm to decode the activity. So right now we have this company incorporated named Xsoft that make this kind of a soft photoresist commercially available, that you can even purchase that from our like a company website, that we can enable this large scale brain probe fabrication through chip manufacturing process. We can make this implantation compatible with small animals that allow us to long-term stably capture activity from the same neurons from the freely behaving rodents. We also partnership with Taiwan Semiconductor Manufacturing Company to build those chips with high density recording channels. Right now, we really solve this kind of a bandwidth issue, that we can build this kind of a soft and flexible electro array at a large scale, like the flexible neural pixel. But instead of like only capture high density neural activity at once, we can capture those neural activity from the same neurons over not just like days, weeks, but months to even years. Recently, we finished the first like a human clinical trial that we can safely implant, deploy those soft electronics into the human brain. Like for the major difference between human brain and the rodent's brain is that the human brain involve a huge amount of the mechanical movement. It's like they are beating like a heart. So that if you use any like a rigid probe getting them into the brain, you will lose the signals like instantly. But by using ours like a soft high density electro array, we can very stably capture the cellular activity at a single cell resolution. And we can correlate those single unit activity to the behavior or sensory input that the human patients have received. So this is just one direction that we're trying to push, that is really use this brutal force to significantly increase the number of the sensors that can be safely and stably deployed into the animal and the human brain. But if you know how those neural activity like connected like between each other, you will feel like when the brain get function matured, no matter how soft and how flexible you design the probe, as long as you implant them into a mature brain, you will break those neural connectivity that cannot be avoided. So this is the like a 3D reconstructed imaging from the TM imaging of the neuron network. In this very tiny region, you see this kind of subcellular regions that is over. There's like a subcellular structure from over like hundreds of the cells that are interwoven together. For the matured brain, the neuron really innervated with each other at a nanometer resolution. So how to address this acute damaging is another direction that we are pursuing to further distribute the sensors across the 3D brain with minimal invasiveness. And then turns out that the nature already kind of gave us a solution. So over the time course of the brain development, we found that really this 3D brain started with the 2D stem cell sheet named neural plate in the embryo, in the animal's embryo stage. And this neural plate will fold into a neural tube with the further growth into like a 3D brain. So we are thinking that in addition to just like a fabricate the flexible electronics, we can fabricate this highly stretchable mesh-like electronics. And then we integrate them with the neural plate. The neural plate folded into the neural tube could like help us internalize the electronics into the brain, and with the further growth and folding, it can help us distribute the sensors across the 3D brain through this like a minimal invasive like a process. So we start to test this idea by using this stem cell derived organoids, because the stem cell derived organoids follow the similar kind of development state, the step as those kind of organs in the in vivo. That is, they start with a 2D stem cell sheet and start to grow into the 3D organoids. And we can design this stretchable mesh nanoelectronics integrated with the 2D stem cell layer, and hope that this kind of morphogenesis process can distribute the sensors across the 3D organoids without damage these organoids. So we start to build this kind of first like a generation of the stretchable mesh electronics, which is a very simple structure with this ultra thin mesh-like structure. We replace this kind of straight interconnects with this serpentine-like interconnects, which has been well developed by many people like Professor John Rogers in the Northwestern University. You can see how stretchable they are. This is a stretchable mesh electronics that is released in the water, that even the floating of the water can easily stretch them. And then we can integrate them with the 2D stem cell sheet. And due to the cell-cell interaction, the stem cell gonna shrink into a thicker cell plate. Actually, this process also help us to further package the electronics into a much higher density structure. And then after 3 days culture, they start to fold into this ball-like structure, start to assemble the electronics in 3D. And after 20 days of culture, they grow into the 3D organoids with all the electronics completely unfold and distributed across the entire 3D volume of the organoids. And we call this technology cyber-organoids. We can basically induce them into whatever organoids that we want. For example, we can induce them into this cardiac organoids. You can see this 3D seamlessly coupled stretchable mesh electronics can accommodate this mechanical contraction of the cardiac organoids. This is allow us to capture the single cell or cellular level field potential across the 3D cardiac organoids. And stably over time, even can help us to illustrate how those signals gradually evolved over the time course of the cardiac maturation in the in vitro setting. Started with this calcium-like electrical activity, gradually merged hyperpolarization, and ultimately generated depolarization. We can induce them into the human brain organoids and capture when the action potential first is generated from this human brain organoids. And in the end, we can implant them into the real living animal. We start with this kind of frog embryo because over the time course of development, this frog embryo is completely exposed to the solution, which is help us to manipulate and integrate the electronics with their neural plate. So this by matching the mechanical property of the stretchable mesh electronics with the embryo, which is much softer than the organoids, we employed those kind of a soft fluorinated elastomer as a dielectric material for this stretchable mesh electronics, and then integrated them with those neural plate. And you can see this neural plate fold into the neural tube will naturally internalize those electrons. And after like a couple of days, development, when they grow from the embryo into the free behaving tadpole, those electrons are already intrinsically embedded into their 3D brain. And this kind of implantation and minimal invasive implantation process allow us to stably capture those brain level electrical signals throughout the entire development. And it's very interesting. This at this neural plate level, we can measure this global synchronized but slow activity, and the activity gradually decoupled into those brain region specific fast calcium-like activity, and ultimately generate this kind of a single unit action potential, and which allow us to really map how those brain activity evolved over the development. So this is this tissue level soft electronics plus the cyber engineering really allowed us to distribute the sensors across a 3D brain tissue. Then the last part is how we integrate the cell type specificity into our electrical recording and control. So over the course of my career, I have tried so many different methods. For example, we tried the optogenetics method that by incorporating the micro LED with our mesh electronics, so that it enables the optogenetic stimulation and the electrical recording, help us to identify what type of neuron that we are recording. Or we use the genetic engineering methods to genetically modify the neurons for them to grow a conducting polymer and link with the electrode and hopefully to capture the activity from the specific group of the cell. But in the end, the method that I like most is to integrate this kind of imaging-based in situ single cell RNA sequencing or spatial transcriptomics with our like a flexible bioelectronics. I'm not so sure if you are familiar with the spatial transcriptomics. I just give a very quick like overview. So the idea is that once you have a piece of the tissue that already finished the measurement, you can like perfuse those kind of hydrogel monomers into those kind of tissue and allow the hydrogel monomer cross-link into a piece of the hydrogel, which will also cross-link those large biomolecules in situ. And then you design those kind of like a procedure that removes the phospholipid bilayers of the cell that enable the optical transparency of the tissue as well as the accessibility for those like large biomolecules that diffuse across the thick tissue. Then we can design the DNA hybridization probe that can be hybridized with those kind of complementary mRNA that we want to identify in the in situ situation. And then each like a DNA probe, we have this kind of a unique gene identifier which can be read out by those kind of multiple round of imaging-based sequencing. So this is the raw imaging of the imaging-based sequencing through just one round of the one round of the imaging. And ultimately, we have the multiple round of the imaging and which combined together can tell us the identity of the DNA and their spatial location so that we can infer the mRNA identity and their location. And then through this multiple round of the cell segmentation and RNA calling, we can assign for each cell with their mRNA copy number. And then it's just a standard bioinformatical process where we can classify, we can cluster the cells at its like a different like a cell type group, and then map it back. We can generate this kind of a 3D cell type mapping. And through this method, unlike those other methods I mentioned, like optogenetics or genetic engineering, we are not just looking at like 3 to 4 genes. Like we can look at thousands of genes at once, so that gives the most comprehensive like a spatial cell type mapping. Then we design this process we call the in situ electro-sequencing. That is after by using our like a soft and flexible electronics to integrate with the tissue. After measurement, they can seamlessly integrate with the tissue into one hybridized structure, so that we can make this entire electronics tissue hybrid structure transparent and directly look at the gene expression from the cells that form the contact with the sensor to identify their molecular identity. Then the real question is that how we know which sensor channel that we are looking at and correlate it with the cell. To address this, we designed this so-called electronic fluorescence barcode. It's a piece of the polymer with this kind of a unique like a structure that is indicated. For example, in this case, it's really the binary structure that is indicated the channel identity. And this kind of polymers is doped with the fluorescence dye and made it through the photolithographic process so that it enable at least through the multiple round of the in situ sequencing to help us to determine the channel information. So this is the raw imaging of in situ sequencing result. And this like it's like we first can get this fluorescence signal, and then we can do this cell map, the cell segmentation and the cell mapping. And then we can assign those kind of mRNA to the different like cell, and through this kind of dimension reduction and clustering process to group the cells with their different like a cell type specificity. And then we can identify the cell that is interested, that we are interested, and then form the direct contact with the sensor, so that through this way we can integrate the cells like electrical property with their gene expression at a single cell or even or just a cellular resolution. And for the neural system, it's slightly more difficult. That is, as I mentioned in the very beginning of my talk, if we have a high density electrode array, that the one neural activity can simultaneously capture by multiple electrodes, and they will show the distribution of the different amplitude of the signal across the different electrodes. So that using this information, we can infer like a relative location of the cells to the high density electro array and then look at their cell gene expression. And through this way, we can integrate the electrophysiology and the gene expression at a single cell resolution for the neural system. And what we are doing by using those technology. So the first one that we can enable this multimodal cell state identification. As I just mentioned, like over the time course of the development at different development stage or maturation at different maturation stage, the cell will show the different like electrical property as well as different gene expression. And for the traditional methods, if we just use the gene expression to define their cell state, well, at the early development state, the cell state can be easily separated with the cells from the resting development state. But at the resting developed state, because their gene expression differences are so subtle, you cannot further separate them. If we just use the electrophysiology to define the cell state, they can be separated into a two big cluster, but within cluster, you cannot easily separate them. But if we combine both like a cell gene expression and the electrophysiology together, we can immediately like separate the cell class cells into those like a very distinguishable like a cell state and correspond well with their like a real development stage. And then this kind of a paired measurement also enable us to look at the correlation between different modality. We can design this very simple one neuron, one layer like artificial neural network structure, like a so-called sparse reduced rank regression analysis. You can unsupervised select the electrophysiological features and look at the correlation with the different genes throughout the development. And just one step, it can tell us what kind of gene regulation change give rise to the different electrophysiological features. So this statistical model is super powerful because for the traditional biophysics model, if you want to model like the gene expression control the electrophysiological features, you can only look at the ion channel genes. But use this like a statistical model, we can incorporate, for example, the structural or metabolic and so many other genes to this kind of to identify their correlations with the electrophysiological features. And with this kind of initial identification of the correlation of the different modality, then we can further design, immediately design this kind of AI-enabled cross-modality prediction model. So this is a model named the coupled autoencoder. Basically, they learn the correlation between the different modality that enable the cross-modality prediction. For example, use the electrophysiology to predict gene expression or vice versa. This is quite important because if you want to measure the gene expression at this large scale, which is actually a terminal measurement, you have to sacrifice the tissue and look at the gene expression at a large scale. But electrophysiology, on the other hand, can be measured continuously, especially by using those flexible or stretchable electronics. Even during the tissue development, we can measure the electrical activity stably over the time. Then we can put those kind of stably record electrical activity into the model. Then you can directly infer like a molecular trajectory of the tissues. And we can even do it like in an opposite way that we know for like a certain patient-specific cells that they have this large gene expression variations compared to the control. We can put those kind of gene expression variations into this model. And you see that you can directly infer or predict this abnormal electrical activity compared to the control and show the remarkable similarity to this real recorded data from those like a patient-specific cell-derived like a cardiac tissues. Uh, in nowadays, in the era of the AI, I think everyone agree that it's not just the data that is valuable. This model that is trained by those like a multimodal data are also valuable. We can design this multitask learning network to learn the correlations between the different modality and enable the cross-modality prediction or enable this kind of multimodality like a clustering methods. And then after this training, we can use another network to directly interpret this like a pre-trained network by like perturb the connection of those artificial neurons in this pre-trained network. Just one step, it can tell us within specific group of the or specific type of the cell, what's the correlation between different gene expression markers to the electrophysiology features or even different morphology. So you say that we build those tissue-like nanoelectronics to enable stable and long-term interface with cells in the biological system. We build this soft electronics, tissue-level soft electronics, and cyber engineering to distribute the sensors across the 3D tissue. And we develop this in situ electro-sequencing that enable the integrate the cell gene expression or molecular information with their like electrical behavior. We kind of like merge electronics from the molecular through the cell to the tissue across all the scale. So what we are doing right now by using this platform technology, the first direction that we are working on that we think that it's gonna revolutionize the entire field of the brain-computer interface. For the limitations for the traditional brain-computer interface is really at the front end. By using those rigid probe implanted into the brain, as I mentioned that due to this mechanical drifting as well as those kind of immune response, even you can stably record the activity from the neurons, you won't be able to record the activity from the same neuron stably. So every day, basically, you are record activity from a different group of the cell. So the focus of this field is on developing how to develop the computational-based like a stabilizer, the pipeline that can do the recalibrate of the cell activity every day. So one of the very famous direction that called the manifold-based stabilizer. So how they do that? If you consider one neural activity as one vector, in the end of the day, depend how many neurons that you record from the system, it will form this kind of n-dimensional neural space. But we know that because the neurons are actually connected with each other, so that they cannot random fire. Their output cannot be those like random dots, like fulfill the entire high-dimensional neural space. It has to be a low-dimensional geometry from this high-dimensional neural space. This is called the neural manifold. And for a lot of people, they believe that for certain specific brain regions, such as motor cortex, this motor behavior related neural manifold are always stable. And they're never going to be changed, even those like a neural system like go through the different experience or memory process. So that they believe that even you cannot record activity from the same neuron, you always can find a stable neural manifold to decode the neural output stably. But we don't have this issue. By using the like this like a flexible electronics that we can capture the activity from the same neurons, this behavior, this motor movement dependent like a neural manifold, or this even lower dimensional structure like a neural trajectory, always stable over this long-term implantation. And even we can use this like a stable recording as a training data to directly train a deep learning network as an animal-specific brain decoder. So this is quite important because in the reality, there's no 100% yield of the experiment. You always have a one neuron that is died for no reason, or maybe another channel that is broken. And for this kind of adaptive animal-specific brain decoder, when this happens, when we have this unit loss for the no reason, that it can quickly be retrained and re-stabilize themselves and give a stable behavior decoding output. And this is just incremental. The real innovation comes from that when we want to move beyond the motor-related brain-machine interface. If we want to decode other information, like olfactory information or visual information, then we'll immediately face this kind of second type of the challenge that named the neural representational drift. So this time it is not just a sensor that is changed, that is sensor is drift. It is that the intrinsic neural dynamics that is changed. Like our brain is a highly dynamic system. Even you don't do anything, you're just daydreaming. That is the neural activity constantly changing within your brain. For example, people have this like animal, like the mice watch the same movie again and again for a long time. And they found that at a single cell level, the cell activity that represent this like a frame of the movie that we call it the neural representation, constant change and never stopped. And it's not just happen in visual cortex or olfactory cortex. It basically exists in virtually any kind of the brain regions other than the motor cortex. And then we found that our technology seems to be ideal to address this question because, as I just mentioned in the very beginning, that we use the waveform that we capture from each channels to determine if we can capture the activity from the same neuron. Then we can look at their like individual neuron representation or the population dynamics. This is a result that we have the animal watch the same movie again and again over like 5 months. You can say that from this kind of recording signal analysis, we can capture the activity from the same neuron over this 5 months of the recording. Then you see that this is really this kind of neural single cell level neural representation over this 5 months of this animal behavior related activity recording. At single cell level, indeed the neural representation constantly change and never stop. And very surprisingly, the change rate is not related to how long they have been watching the movie, but only related to how many times that they watch the movie, and they never gonna be stopped. Then the question is that because we know that the animal is watching the same movie again and again, then what is really stable? What is really stably like preserved or perceived by this animal's brain? Then if we're not looking at the single cell level dynamics, we look at the population dynamic, we look at the neural manifold, we found that the neural manifold, when we can capture the activity from the same neuron, seems to go through that mathematically called affine transformation. That means that they preserve their topology. For example, if the manifold starts with a donut-shaped structure, this donut-shaped structure gonna like rotate, translate, stretch in this high-dimensional neural space. But they are always gonna be a donut-like structure. So that then after we know this is a affine transformation, then things become easier. We can design another neural network to compensate this affine transformation. Then we can give get a stable visual information decoding. Then this is really like a spark me for another thinking that why brain have this constantly like a changing behavior, constantly change the dynamics because it seems to be like an energy consuming process in contrast to our like a thought that the brain should be like a energy reserving like a system. But then my colleagues in the computer science told me that in the field of the AI, there's actually the big question, big challenging for those like modern deep learning neural network that is called the catastrophic forgetting. So what does that mean? If you for example, if you have a small network, you train it to identify a cat. Then after it learn how to identify cat, you train it to identify dog. Then they're gonna quickly forget about how to identify the cat. Then we are thinking that maybe this kind of brain dynamics, this intrinsic neural representational drift, represent a mechanism that mitigates this catastrophic forgetting, that enable the continuous learning capability that our modern AI system lack of. So how that could work, our hypothesis that after they learn this dynamics of those kind of initial tasks, this learned dynamics gonna constantly migrate or drift inside this kind of low dimensional neural space. So that when this new information that has been learned, it will not quickly overwrite the previously learned information. So to test this hypothesis, we design a new type of the deep learning architecture, which shares the same like architecture as a traditional deep learning. Instead, that we introduce the synthetic noise into the system that allows the learned dynamics cannot form a stable local minimum, but instead that it will constantly migrate in this loss landscape of the deep learning network, that when the newly learned information, it will cannot overwrite the previously learned information easily. So we test this idea on the different kind of neural network and ultimately on those kind of large language model. For example, in this case that you can say that for a stable neural network, deep artificial neural network, when they learned the new task, it will quickly forget the previously learned information or previously learned task. You cannot maintain its performance. But when we introduce this kind of a constantly drifting mechanism into the deep learning network based on the information, based on the dynamics that we observed from those kind of brain dynamics, it can seem to learn the new task without lose the performance of the previously learned task. So we think this is really interesting that if you imagine that the previous like a measurement system, you have this kind of neural intrinsic dynamics that the neural activity constant, neural representation constantly change. And then you have this recording instability that the interface with the neural system also constantly change. So the traditional technology won't be able to separate or decouple them at all. Right now, we can fix the recording from the same neuron. So it gave us the opportunity to really observe this intrinsic neural dynamics. And what's more is that we imagine that we are building this AI agent system that is can be completely programmed, reprogrammed by those like a neural representation dynamics. And we can connect them through this flexible electronics that when the animal is conducted different type of task like memory learning, it will reprogram this AI agent. And after this process, we can use another like a interpretable learning mechanism to perturb or to understand, explain this AI network that has been trained by the neural dynamics to really understand, to decode the brain intrinsic dynamics. The second direction that we are very interested in is stem cell engineering. So there's one major challenge in the field of the stem cell biology or stem cell based therapy. That is how to quantify the function maturation of the stem cell derived tissue, especially the stem cell derived organoids. That we want to say that we can using the technology that to stably capture the electrophysiology from those tissues. And then we can do the dimension reduction actually to analyze this kind of continuous measure signal to define or benchmark their development. So then we tested this idea started by working together with Professor Richard Lee in the Stem Cell Institute because they previously found that when you integrate the endothelial cells together with the stem cell derived cardiomyocyte, it can mitigate its electrical the functional property. It seems to make them like become more functionally matured. And the previous like hypothesis is that because the stem cell derived endothelial cells will quickly form this three-dimensional vascular structure, which help those nutrition and oxygen diffuse across the 3D tissue. It will help it like a functional maturation. Then we using our system to test this idea, we found it's actually not true. That only just after like 1 or 2 days like integration of those cardiac stem cell derived cardiac tissue with the endothelial tissues, the activity of the cardiomyocyte already be like mitigated. And then if we analyze those kind of like standard like features from the cardiac like field potential, we found that there are significant like improvement of the amplitude or the DVDT or spike duration. And then we borrow how then the question is that how we quantify this, right? So we borrow the same idea that have been previously used for the single cell gene expression analysis that called the pseudo-time analysis, which is basically just a dimension reduction methods of this high dimensional data, projecting them into a low dimensional space and look at the correlation between them. So we apply this pseudo-time analysis and to this high-dimensional electrophysiological data, and it works remarkably. That is help us to align this kind of trajectory of the electrophysiological development throughout this kind of functional maturation. And then we put things like together. We found that it's not really just not for this long-term like interface. It's just this day 15 is like how long the cardiomyocyte has been in vitrally developed or differentiated, and only after like 2 to 3 days incorporated with endothelial cells. But just like this 2 days integration, they strongly shift the functional maturation stage of the cardiomyocyte towards a more matured stage. And then we can, using our technology, look at the gene expression at the specific like development stage that we want that shows the drastic like a difference of the electrical maturation stage. Then it's like really tell us, the result really tell us it's really coming from this cell-cell interaction that strongly mitigate the functional maturation stage of the cardiomyocyte from the endothelial cells. The results actually show a very interesting the result that is due to this kind of ligand receptor interactions, and then over the time endothelial cells become more like a cardiomyocyte, and the cardiomyocyte cells become more like endothelial cells that mitigate its functional maturation in this intact piece of the tissue. Then that's the next question that how we use the system to further improve the functional maturation of the stem cell derived organoids. Then it's become like the question of the algorithm that how we analyze the electrical recording that we directly from the tissue, and then generate a control policy to mitigate the cells like throughout this development. So in the era of the AI, especially if you play, if you look at the AlphaGo, the mechanism of the AlphaGo is that we really don't need to design, make a predefined like algorithm to determine what is the optimized like control condition, but rather we build this kind of a trajectory system that give the state a very clear state information for the AI agent, and then we just want to point out the final goal and ask the AI agent to figure out to perturb the system, look at the response and figure out what is the most optimized stimulation system. Then we build this closed loop system and using this very simple base optimization to look at how to optimize the two parameters like amplitude and frequency. Then in the initial of this like testing that we go through this multiple batches of sample, each batch of sample like contain like a 3 to 4 organoids with the electronics incorporated. You find that this AI really get confused that explored across the entire parameter stimulation parameter space. But over time, they seem to be converged. They seem to be learned something that given a studio time that from inferred from this like a recording data, they can give a stable stimulation output. And how the result looks like. Then after this like AI driven like stimulation, the studio time of the electrical recording data significantly shifted towards to more matured state. And if you look at raw data, the amplitude of the action potential collected from the cardiomyocyte become much larger. And more interesting is that the batch to batch variation has been substantially reduced. And if you look at the tissue level property, this AI-driven stimulation, they significantly improve the propagation speed of the action potential propagated across the synthetic cardiac tissue. And also at the molecular level, the cells seems to form like more aligned molecular structure in the tissue that receive this AI-driven stimulation compared to the traditional fixed stimulation or this kind of no stimulation control. And in the end, that we are very interested in, we are really interested in find that we can actually even capture the activity from this like stem cell derived pancreatic system. That because this like a pancreatic organoid is probably the only type of organoid that have been used clinically right now. So when like then like my colleagues like Professor Douglas Melton and his previous postdoc then right now is like a professor at UPenn introduced me the concept that the pancreas actually derived or developed from the same group of the cell as the brain development. And at that time, I even don't know where the pancreas is. So but they try to convince me to incorporate our electronic system into their like a stem cell derived pancreatic organoids and capture how those like, kind of electrical property change in the pancreatic system. Then we found that we can measure this beautiful action potential like activity from this stem cell derived pancreatic organoids. And more interesting that we can use those spike sorting algorithm to actually sort those kind of spike from our measurement result and group them together into the cell specific activity. And then we can look at the different cell cluster response differently to the glucose thresholding to tell us with the like a pharmaceutical confirmation to tell us what is the signal from alpha or beta specific activity. Then we can look at how those activity evolved over the time course of the development of the pancreatic organoids. We found that we can actually further identify the different type of the electrical two different electrical state from the alpha and the beta specific cells. Like one state that show much less basal activity for the alpha and the beta specific cells. And it's really like over the time course of the in vitro maturation, it's the ratio of the different type of the alpha and the beta specific subgroup cells change and make the whole tissue more function mature. And we can even further like using test a different protocol to enhance the function maturation of the pancreatic organoids, such as using this feeding instrument that we also can measure this periodic like activity from this electrical recording of the pancreatic organoids, which seems to further improve the like sustain the function maturation of the pancreatic organoids in the in vitro setting. And the same thing that we can look at their gene expression at a different development stage or maturation stage and tell us what kind of underlying regulation that gives rise to the different functional maturation state of the cell. And in the end, I just want to very briefly introduce the functional brain atlas that we are trying to do by using our technology. By using the spatial transcriptome technology, we already map the like a 3D brain's gene expression at a subcellular resolution. And it's a really like a very interesting result that we can even like redefine the brain anatomy by the single cell gene expression rather than it's like traditionally defined by the morphology. And it's not just like our lab is doing that. I'm sure you know that there are so many labs like in Cambridge or globally are working on the same thing. That in 2023 that we published 10 papers at the same time on Nature by using the different technology to look at the cell gene expression across a 3D brain. And my student built this like a very beautiful foundation model by leveraging the cell gene expression and their spatial location to integrate this one centralized atlas, brain atlas, contain over 18 million cells, which you can also incorporate the different modalities through this kind of multimodality measurement, such as we can use the flexible electronics measure this behavior-dependent electrical activity, harvest the tissue together with the embedded electronics, look at their gene expression, and then incorporate those kind of electrical activity back to this centralized atlas through this spatial gene expression mapping. Then the only challenge is that how we can analyze this like a drastic amount of the data from those kind of measurement system. So that we are currently very interested in building this large language model driven AI agent, this kind of AI workflow that enables autonomous data analysis as well as this multimodal, especially visual language model based data curation. But it will help us really to incorporate everything into one big centralized atlas. And then we want to use this multimodal data to train the AI agent and then in the end dissect the AI agent to tell us the correlation between different neural activity cell type and the animal behavior. We hope that we can build this brain functional atlas, really function like a Google map of the brain to tell us the further target for the brain recording and the stimulation. So that will be the end of my talk. And I want to disclose my conflict interest. I will be co-founder for not just the Xsoft, but right now it's several companies, and we're heavily involved in the human clinical trial study that to test this chronic brain implants used for the brain-machine interface as like a neural modulation. And this work is mainly done by fantastic group